Hello, hello. It's been a couple of days since the last time I sat down to record a video. I do remember that the last thing we talked about was transcriptional control in prokaryotes. We went through a description of the lac operon and to a certain extent the trip operon. I only included the trip operon there as an example of a repressible mechanism, just to kind of contrast it with what was going on with the lac operon. But with the lactose operon, I gave you this idea that we've got three genes, we need them together, and we need these three gene products together. Why not just flip one switch and make all three gene products happen? And that's how we did it. We had a single control region on the lac operon. When the lac operon got turned on, we started making the one long messenger RNA molecule. From that one long messenger RNA molecule, we ended up with three different proteins, three gene products: lactose permease, beta-galactosidase, and galactose transacetylase. I mean, you don't need to know the enzymes, but you need to know that these are the three enzymes that we need in order to use lactose as a food resource if we happen to be E. coli cells, right? And lactose is present, right? And so I guess the main theme there was, the operon structure was important, but the main theme there was that we want to turn on the genes when the genes are needed and not when the genes are not needed. I did mention, I gave you this slide. They mentioned that in eukaryotes, we don't have that same operon structure. If we've got three gene products that are needed, if we need these three genes, lactose permease, lactose, beta-galactosidase, and galactose transacetylase. I mean, if we need those three gene products, we're going to be making three separate mRNAs, okay, from three separate genes, and each one of those transcriptional units is going to have only a single, it's going to have its own on-off switch. Okay? And so when we think about it here, okay, you can say, well, if it was an operon structure, annotation, operon, prokaryotes, it's really efficient. And comparatively with eukaryotes, it's simple. I know it didn't seem simple last time when we talked about it, but yeah, realize that if you have just one on-off switch for a whole bunch of genes, that's a simpler system than having multiple on-off switches for each gene, or multiple on-off switches, one for each gene, right? And in eukaryotes, the eukaryotes, the control is far more complex. The different pieces and all the bits that have to fit together, and you have to have exactly the right constellation of transcription factors. It's really a lot more complex, and you might say it's less efficient. And I guess it is, right? But at the same time, it does allow for greater flexibility, which in the long run actually makes efficiency greater. And by that, I'm going to skip all the way down to the very bottom. It's going to be the last thing we talk about today. And this comes from something we learned from the Human Genome Project. And when we first engaged in the big task of sequencing the entire human genome, this would have been in the 1990s, people were taking guesses as to how many protein coding genes there would be in the human genome, right? And you take stock of all the different things that have to happen with different kinds of enzymes, different kinds of proteins in the human body, and you're really looking at a minimum of 100,000, maybe a few 100,000 different genes. And so imagine our shock when the number came out substantially lower than that, not just substantially lower, way lower than that, right? So if we thought that the minimum threshold for the genome complexity would have to be about 100,000 genes, and we find less than 20,000 protein coding genes, that's a pretty huge shock, right? And so that's, I mean, if we're actually getting all of that bang for our buck with only 20,000 protein coding genes, you might say that, well, that's the ultimate in efficiency, right? And so I guess one of the themes that we come to with today's lecture are how many ways in which we can account for one set of a relatively small set of 20,000 genes, allowing us to create the complexity that would otherwise be expected to have a much greater number of proteins, protein coding genes. Okay, and so I'm going to start you off here. Now we're going to be looking at the actual mechanism that's required in order to initiate transcription, right? So this would be the equivalent of having the repressor removed and having the promoter attract the RNA polymerase, and the RNA polymerase starts chugging down the gene. But in this case, that control region is going to be far more complex, and it's going to require a lot of pieces. And your book shows it this way. It's actually a really nice diagram. And so these things that are in the piece, in the pieces in the puzzle, that's what I was trying to say. Pieces in the puzzle, including little regions in the DNA. Right? These are the little, they're actually in the code. They're not separate units. These are little stretches of DNA that run upstream from the actual gene. The gene is down here, right? The gene goes in that direction. We're gonna have to transcribe all of this. This is what gets converted into an RNA molecule, an RNA transcript. This is our RNA polymerase here, but these upstream regions that will interact with the transcriptional upstream region, they might be. This is drawn the way this is drawn. It seems like it's only a few spaces down on the DNA, upstream on the DNA. Okay, but the real truth is that this might be several 100,000 bases away from the actual gene. Right? There's one situation that I know of in which this gene over here, okay, requires an enhancer unit that is several genes, right? There's all of this DNA that's come in before we actually get to the part that will have the enhancers, these enhancers that are required in order to make this loop. Okay, doesn't require us to make a small loop like this. Sometimes the loop might look incredibly large, right? And so yeah, that's another dimension to the overall complexity of the system. But in addition to these enhancers, we're also requiring activator proteins and transcription, other kinds of transcription factors. Sometimes there might be a DNA bendy device, and that's all required. This all has to fit together. These are puzzle pieces. Come together like a jigsaw puzzle, and they fit together and they create this little complex transcriptional thingy that will bind to the enhancers, result in this little crook, and that's where you get the attachment of the RNA polymerase. RNA polymerase then will come in and start the process of transcription, right? And so that's basically the mechanism. I'm not really that concerned about the mechanism. I think what I would really like to return to is our theme of how this system actually is adaptive. Why would we want to do things this way when bacteria have this really simple way of turning a whole bunch of genes on in the locations where they're needed? Okay, and so I'm going to go back to my original share whiteboard. And I'll be giving you a similar drawing here. And what I want you to think about is, well, how could having a system like this allow for, let's make sure I'm drawing the right color. Okay. Allows for precise control of gene activity in some cells, but not others. Okay. So I guess the question is, turn this gene on in some cells, but not others. And this is particularly important in a multicellular organism like a human, right? And so, there might be some genes that would be required in liver cells and tonsil cells, right? But not in kneecap cells, right? And so if these are our three transcription factors, These are 3 transcription factors. And the 3 pieces that we need to put together in order to initiate the activity of this gene, right? This is the gene, gets transcribed. Let's say we need to have, see if I can do this right. If the transcription factors are I, C, and P, right? ICP. And so if these 3 things are required, these 3 transcription factors would be present in liver cells and in tonsil cells, but not in kneecap cells, right? So, kneecap cells are missing the I, they're missing the P, and so kneecap cells would not have the appropriate combination of transcription factors required to turn on this gene. Right? Maybe there's another gene, a different gene, and this other alternative gene. Maybe we don't need I and C and P. We're going to need A, B, C, and D. Okay. And if the transcription factors that we need, there are actually 4 of them, right? So there are 4 transcription factors, A, B, C, and D. And so if those are the transcription factors that are required in order to turn this gene on, because that's what is necessary in order to make this little crook, what kinds of cells would be expressing this gene? Okay, that was easy enough, right? You looked over here and said, well, liver cells have A, B, C, and D. Kneecap cells have A, B, C, and D. So the correct answer is that this gene requiring the transcription factor puzzle to include pieces A, B, C, and D, it would be a gene that would be active in liver cells and in kneecap cells, but not in tonsil cells. Got that? And so you realize that the actual mechanisms for transcriptional control are far more complex than this. It's I gave you kind of a simple idea. We have thousands of different kinds of cells in our body. We've got nearly 20,000 genes that are going to be turned on and off in different subsets of the body. So the actual mechanism is going to be more complex than this. Not that much more complex. There might be as many as a dozen transcription factors that are required. But if you could decipher the appropriate combination of transcription factors, you would be able to turn a gene on based on that factor alone, on that ability alone. Okay, so let's go back to our previous share. No operon structure, you got that. There are a lot more pieces to to control the transcription of a gene, regulatory genes. Oh yeah, and by the way, all these transcription factors this is sometimes a little bit confusing. The things that we are identifying as transcription factors or activators, these are proteins. These are actually proteins, but not the proteins that our cells need, like enzymes and membrane proteins, but not the proteins that are included in that 20,000. That number 20,000 I gave you before of the number of protein coding genes. We're not including the regulatory proteins, the transcription factors and activators, and all the other things that might be there, because we haven't really characterized those well enough to include them. Right? There are a lot of other regions of DNA that are important for transcription, like the enhancer that we showed you. There are also silencers that can effectively turn genes off if they have the appropriate set of transcription factors. Yeah, transcription factors can interact with regions of DNA, not only to activate genes, but also to attenuate them, to turn them down. Right? And these these transcription factors may be off switches in the way that a repressor was an off switch, or on switches in the way that you could turn on genes, as we did in the case of the lac operon. Okay, so I've done all parts of 1. Now we need to go into item number 3, course control of transcription. And before we actually get there, I want to put a little reminder way back in Unit 1, when we're first struggling with chemistry. We were thinking about different kinds of functional groups that were there on organic molecules. Maybe you'll remember this. If we have an organic molecule and there's an OH group on it, we call that, that's right, hydroxyl. And you might remember that a hydroxyl group contributes to the hydrophilicity. It makes things more water soluble or more water loving. If you only have hydroxyl group, it makes an alcohol. We learned about amino groups. We learned about carboxyl groups, carboxyl, and a few others. And I promised you that there were a couple of organic functional groups that we would be coming back to much later in the semester, and here we are, right? And so the groups that we're looking at now are methyl groups and see if I do this right. And acetyl groups. Right. Now, don't worry about knowing what they look like. Okay, I might have even gotten these wrong. And if I got them wrong, yeah, shame on me. But it doesn't really matter for us. You need to know the words methylate and acetylate. Basically, if we methylate something, we're adding methyl groups onto the molecule, right? A molecule might look like this initially, and then we're going to be coming along putting a CH3 on it. That would be a methylation, right? Alternatively, the molecule might look like this, coming over here and putting an acetyl group on it. That would be acetylation, right? And these are the modifications. These are the chemical modifications that are being made either to DNA, because if we have a cysteine residue, or sorry, a cytosine residue, I'm confounding a whole bunch of things, right? Cysteine is an amino acid. DNA cytosine is one of the 4 bases that might be there. Cytosine might become methylated, or the histone protein that is used to pack DNA to create chromatin from DNA, right? Chromatin is a combination of histone proteins with the DNA. If we acetylate the histone proteins, that's another kind of modification that's going to be occurring. So maybe this would be a cytosine, this would be a histone, and that's the way the kind of modifications we're talking about are going to work, right? And apart from that, it's really basic. Let me add one more thing. You know that DNA is double-stranded, right? But if we kind of simplify a DNA molecule, it's really thin. We have the other piece, which is the histone protein. Histone proteins are coming in these little packages, and the DNA does this really cool thing, which is the wind. So we end up with these little balls of DNA that are wrapped tightly around histone proteins. And then this structure gets packed even more tightly into this kind of wet, windy DNA. So these are all packs of DNA histones, and histones might either be relatively loosely packed, as I've drawn it here. In other words, you can see there are little spaces, and you can see in between here. Or alternatively, you could have like super densely packed. Chromatin. So So dense, loose. Okay. You know, these are the two ways that the actual chromatin might appear in a chromosome. It gets even denser during mitosis, but we're talking about during the regular gene activity parts of the cell cycle. You might have relatively loose chromatin, you might have more dense chromatin, and the loose one tends to be more active, right? Because kind of makes sense. Because if the DNA is packed relatively loosely, that's when the transcription factors and the RNA polymerases can penetrate in there and actually activate the genes that need to be activated. Okay. Whereas if things are so tightly packed that we don't really leave any space for any of those other particles to go in and interact with the DNA, dense DNA is pretty much turned off. And that's what's going on with methyl groups. So methyl groups, you attach methyl groups, this is going to be causing dense packing. In other words, tending to turn genes off. Whereas when you acetylate the histones, that tends to make the DNA pack even more loosely than this. And so this loosens chromatin structure. So, and it allows the transcription factors to enter. This is going to make things more active. Yeah. And so we can completely shut DNA off with very tight packing, and we do that with super heavy methylation. We can do it slightly with a lower degree of methylation. We can make DNA chromatin pack more loosely with acetylation, or very loosely with an even higher degree of acetylation to the histone proteins, right? And so this is kind of like, I think of this like a coarse focus knob, right? Yeah, no, you guys are not in the laboratory, you're not playing with microscopes every week like the other classes, but in the other class, we've got a coarse focus knob on the outside and a fine focus knob. The coarse focus knob allows us to get really gross control, whereas the fine focus knob on the inside gives fine level control. You might think of the coarse focus knob as being the methylation, acetylation, right? Whereas the fine focus knob would be the transcription factors interacting with the enhancers, turning genes on precisely when they're needed and not when they're not needed. Okay. So let's go back to our main page here. Chromatin, course control, histone, acetylation, upregulation, cytosine or DNA methylation causes downregulation. Done with that. Now the other thing about this is that once you make these modifications, once you methylate the cytosine, if this is a cell in which you're doing the methylation, right? Turning genes off in this cell. When this cell divides, right? In other words, when the DNA gets replicated, if you have the DNA methylation, that methylation gets passed down to the daughter DNA, right? So methylated DNA will give rise to methylated DNA. If the histones are acetylated, that will also get passed down, right? What we call these epigenetic modifications because they're kind of like on the surface of genes, right? The genes are on the DNA, and we're making changes to the genes or changes to the histone proteins. And these epigenetic modifications are passed down from one generation to the next. So this gets to the idea that once you turn off a certain region of the gene in development, it is turned off permanently. We're not going to be able to erase those epigenetic markers because generally speaking, when we replicate the DNA, those epigenetic markers get passed down to the daughter cells. Okay. Now there's one place where we need to erase those epigenetic markers. Okay. Okay. And that's when we get to the production of gametes, because the gametes are the reproductive cells. The gametes are going to give rise to the zygote, which is the baby of the next generation, right? The zygote has to have all the genes on again. We can't leave any genes completely permanently turned off. And that's why zygotes are starting off life with no epigenetic modifications, right? Does that make sense? So yeah, I mean, the other thing that kind of goes along with this, and it's a little bit aside from what we're talking about here, but in the process of development, we're going to be turning genes off. As those genes are turned off, that's happening through epigenetic tags that are put onto cells and onto the genes in cells. But at some point, we do have to erase all of those tags. So when baby gets born, is going to be able to put its own tags on during its development. Okay. So done. The last thing we want to get back to is a couple of lectures ago, we talked about how in the RNA structure of eukaryotes, it's another difference between eukaryotes and prokaryotes, is that in eukaryotes, we have introns and exons. There's this interrupted structure. We have exons that get that are actually coding for something. They actually make it into the process, into the final messenger RNA molecule. But between transcription and the actual production of a final messenger RNA molecule, we have to do editing, which means we've got to get rid of the introns. Right? Now, this actually plays into today's lecture really nicely because in addition to everything we talked about so far, it's possible for us to, this is our DNA, right? And in the process of transcription, we're ending up with an RNA transcript. But we also have to realize it's going to be an editing that goes on between transcription and the final mRNA. Okay. I haven't added the GTP cap or the poly A tail. That's going to happen the same way. Okay. And what I want you to think about here is that in this RNA transcript, we've got 4 exons. Okay. I've got exon, I call it exon A, exon B, exon C, and exon D. And then we also have 3 introns, intron 1, intron 2, intron 3. Right? Okay. And in the process of RNA editing, we remove the introns, but it's also possible for us to remove exons as we splice things out. Okay? So what I'm drawing here over on the left, what you want to imagine is that we're going to be splicing out. So this might be the first exon, and then we're going to be splicing out the first intron. There's a little loop. And now here is exon B. And then we're going to be having next splicing, but in this case, we're going to be splicing out not only exon 2, there's exon 2, we're also going to be splicing out, intron 2, we're also going to be removing exon C as well as intron 3. So there's intron 3. And then we're going to be finishing it up with exon D. So we basically end up with A, B, and D. Right? In a different cell, meanwhile, in a different cell in your body, you could do something similar, only in this case, we're going to be splicing together exon A, exon C, and exon D, and remove exon B. So in this case, we would have exon A, intron 1, exon B, intron 2 splicing out, get exon C, then we splice out intron 3. Oops, that was the wrong color. It should be black. A little longer, and that's how we end up with a different messenger RNA. This one contains A, C, and D. Right now, the reason why this is kind of cool, because you've talked about membrane structure and the idea that proteins are going to be associating with membranes. And so let's say that B is the region. Let's envision that B, this is the region that codes for part of the protein that attaches to the membrane. Whereas the parts A and D are the parts that code for the catalytic unit. So in other words, we're able to make two versions of the same enzyme. One where the enzyme is bound to a membrane, right? And two, we don't have that membrane domain in our second mRNA. In other words, we might have something else, but this might be giving rise to a version of the same enzyme. It's exactly the same enzyme. It's coded for by the same gene. But in this case, the enzyme is not binding to a membrane, and it stays in its globular shape and floats around the cytoplasm, right? Okay. So, effectively, we're making two different gene products. Even though we're using the same transcriptional unit, there's one gene. It can be turned on and turned off in different cells, as we know, because of the way that transcription factors are so complex. And also, we can actually make that transcription unit, the RNA that we transcribe off of the DNA, serve different purposes, right? And I've just given you a simple version here. There are sometimes dozens of introns in a gene that could be spliced out in different ways, giving rise to different kinds of gene products, all starting off with exactly the same gene. Okay, and so again, coming back to our main conceptual motif, the idea that we can create many gene products with a smaller number of genes than gene products is an important realization that we've turned to since we've become fully aware of this, since we have the information from the Human Genome Project. Up until then, we were pretty much convinced that would be one gene for one protein. Okay? We know that's not the case. Okay. There's one more thing that I want to share with you. This is relating to the textbook, and it has to do with everything with genome structure. And so I'm going to skip down quite a ways. We'll be looking at chapter 12. And chapter 12, part 18, I have this in the readings. And I just wanted to share with you this diagram because it kind of gets you to appreciate how little of the genome is actually playing along with the rules that we have described so far in BIO110, right? And so if you look at this pie diagram, we know we've got 20,000 genes. Okay? 20,000 genes is a number that I've given you many times at this point, right? And so, like color red, these are the 20,000 genes. 20,000 genes that actually code for either proteins that are important for our normal life, or the ribosomal RNAs or the tRNAs. These 20,000 genes are only about 1.5% of our whole genome. Right? Regulatory sequences, these are the ones that code for transcription factors and other things. You know, we have to have a pretty sophisticated crew of transcription factors making it possible for us to do all the things that we want them to do in the different parts of our bodies. So yeah, I mean, it's a much bigger portion of our genome, okay? But it's still small overall. You know, how do we account for the remaining 93.5% of our genes, of our genome, right? Not genes, because they're not genes. If these are genes. These are, I guess, genes for transcription factors. And they're not all genes for transcription factors. There are other things like the enhancers and the activate. You know, the activators are actually proteins, but other portions of the gene that might have some role in controlling DNA. Okay. Introns, look, there's way more intron DNA than exon DNA, right? And then the remaining of this would include, well, it's one group called transposable elements, which are like parasitic bits of DNA. It's kind of scary, you know, these little stretches of DNA copy themselves and make themselves more abundant in the genome, and we just have to live with it because there's not a whole lot we can do. There's repetitive DNA that we don't recognize as being related, and there's DNA that doesn't seem to serve any purpose, that we can't find any relationship with either existing genes or with the parasitic bits, right? And so yeah, the actual portions of the gene that code for our cells and the parts of our cells is really pretty minor. It's the part that the things that you're made up of requires far less DNA than what you actually have. Okay. Do all organisms have such a huge amount of, is it wasted or junk DNA? And it's kind of an interesting question. We're coming to realize that maybe some of this junk is not entirely useless. Some of it might actually be pretty important. But at the same time, we have the information from the humble fugu, right? Anybody know what a fugu is? Yeah, it's a pufferfish. But fugus are really interesting because they have very little junk DNA. Their genome is way, way smaller than a human genome, in part because they don't have a whole bunch of this stuff. They're basically getting rid of a lot of that stuff, and that leaves them with a much smaller genome compared to what most other kinds of animals have. Right. Completely useless trivia for you, but that's a good place for me to end this lecture.